Hello everyone, my name is Pingky and I'm here with my partner Nawal. Today we would like to present a paper entitled A Nasal Breath-Based Classifier of Asthma Identified by Machine Learning Analysis of Nasal RNA Sequences Data. Okay, this study addressed the problem of inaccurate and missed asthma diagnosis caused by the limitation of current diagnostic tools. To solve this issue, the researcher identified nasal gene expression biomarker using machine learning. Why this problem matters? First, asthma affects about 8.6% children and 7.4% of adults in the United States. In addition, many people with asthma-like symptoms remain undiagnosed. The second, current diagnostic tools such as pulmonary function tests require specialized equipment and trained professionals, so many patients never undergo the testing. Okay, there is also research gap in the previous studies. First, earlier studies use small sample size and array technology. The second, there is still no asthma classifier based on RNA sequence and machine learning using large datasets. And this study used transcriptomic data in the form of RNA sequencing obtained from nasal breathing. The response variable was asthma status, while the predictor variable were thousands of genes expression level from RNA sequence data. The researcher applied several machine learning methods, including logistic regression, SVM, random forest, and ADABUS, along with recursive feature elimination of RFE for feature selections. Machine learning methods were chosen because the first genomic data is extremely high dimensional and feature selection helps reduce noise and overfitting.
Okay, next is the main findings of the paper. So the key findings of the paper has results. The first one is the AUC score equals to 0.994 and the positive predictive value equals to 1, negative predictive value equals to 0.96 and the F measure equals to 0.98 for asthma patients. Next. So the model demonstrates very strong predictive power when tested on an independent RNA sequence test. As I explained before, the PPP score and the NPV score and the AUC score. So this model outperformed alternative models that include all genes, all differentially expressed genes, or only genes known to be associated with asthma. Next. And the important genes or biological mechanism, so the 46 of the 90 genes in the classifier are genes whose expression level differs significantly in asthma patients, which the 22 genes are overexpressed and the 24 genes are underexpressed, which these genes are associated with important biological pathways such as the immune system, the adaptive immune response, the innate immune response, the defense response, and the response to external stimuli. Next. Okay, the study has several strengths. So the first, it used one of the largest nasal RNA sequence asthma datasets at time, making the results more reliable. So the second, the researcher applied a robust machine learning pipeline, including next class validation, RFE, multiple classifier, and statistical tests, which help reduce overfitting and improve model stability. So the third, the model was validated using external RNA sequence and microarray datasets, showing that it's fairly generalizable and specific to asthma. Finally, the nasal brushing method is minimally invasive and more practical than pulmonary function test, giving it potential clinical value. However, the study also has limitation or weakness. So most samples come from a single center were dominated by Caucasian subjects. So the generalizability may still be limited. So some external datasets were also small and unbalanced, which could affect performance stability. In addition, difference between RNA sequence and microarray platforms reduce performance in some retrospectives. So prospective clinical validation is still needed before real clinical application. Next. So the future research will we do First one is additional data. So recruit larger test cohorts with greater racial diversity across both children and adults to better represent all populations. The second one, the statistical model development. Move beyond simple binary diagnosis, asthma versus no asthma, to predict specific asthma endotypes and treatment response for personalized therapy. The third one, real-world validation. So the next final step for validation is testing the classifier prospectively in large real-world clinical cohorts. The last one is practical or policy application. Develop a rapid, automated, and minimally invasive nasal biomarker test for primary care frontlines, providing an immediate solution for clinicians without access to pulmonary functions testing equipment. Next. So that's all from us. Thank you, everyone.